Welcome to this lecture on the fundamentals of biomolecules. Today, we're going to explore a highly misunderstood but absolutely vital class of molecules known as steroids. When most people hear this word, they immediately think of synthetic muscle builders. But in biochemistry, steroids are naturally occurring rigid molecular keys that float through your bloodstream. They are essential for your survival, unlocking specific doors in your cells to trigger growth, manage stress, and digest food. Let us dive in and understand how they work from the ground up. Now that we know steroids are not just for bodybuilders, let us look at what they actually are, intuitively. Imagine a set of molecular keys designed to unlock specific cellular responses. Other lipids, like the fats and oils you eat, look like long, floppy chains. Steroids are completely different. They have a very rigid and sturdy shape, built almost like a microscopic honeycomb made of interlocking rings of carbon. This rigid structure is their superpower. It allows them to wedge perfectly into cell membranes to provide structural support, or to fit exactly into specific receptors in the body to trigger biological processes. Building on that intuitive picture of a honeycomb, let us define steroids formally. In biochemical terms, steroids are a specific class of non-saponifiable lipids. This means they cannot be broken down into soaps by alkaline hydrolysis. What truly defines a steroid is its highly conserved carbon skeleton. Every single steroid is built upon a foundational core composed of exactly 4 fused carbon rings. Scientists conventionally label these 4 rings as A, B, C, and D. This universal 4-ring structure is the defining hallmark of all steroids, no matter what specific function they perform in the body. Let us look closer at those 4 rings we just introduced. The first 3 rings, labeled A, B, and C, are 6-membered cyclohexane rings. The 4th ring, labeled D, is a slightly smaller 5-membered cyclopentane ring. Together, this intact 4-ring core is known as the cyclopentanoperhydrophenanthrene nucleus, or much more simply, the sterane nucleus. Nature creates different steroids, like cholesterol or testosterone, by attaching various functional groups to this rigid central core. Because this core is predominantly composed of carbon and hydrogen atoms, steroids are highly hydrophobic, meaning they repel water and do not dissolve well in the bloodstream. To truly grasp the sterane nucleus we just discussed, let us look at this diagram. On the left, you see the bare sterane nucleus. Notice the 3 6-sided rings, A, B, and C, fused to the 5-sided ring, D. This is the rigid honeycomb we talked about. Now, look at the right side. This is a complete cholesterol molecule. You can clearly see the same 4-ring core, but it has been modified. There is a hydroxyl group attached to the bottom left, a double bond in the second ring, 2 small methyl groups pointing up, and a long, flexible 8-carbon tail extending to the right. This visualizes how a basic core becomes a specific functional molecule. Now that we can visualize their structure, let us explore why these molecules are so biologically important. Steroids play incredibly diverse and critical roles in almost every biological system in the human body. The most famous, and certainly the most abundant steroid, is cholesterol. While often viewed negatively in popular culture, cholesterol is actually a crucial structural component in animal cell membranes. It acts as a buffer, regulating the fluidity and stability of the membrane across different temperatures. Without cholesterol wedged between the other lipids, our cell membranes would either become too rigid and crack in the cold, or become too fluid and literally fall apart in the heat.
Beyond stabilizing membranes, cholesterol serves another vital purpose. It is the biological precursor for all steroid hormones in your body. This means your body uses cholesterol as the raw material to manufacture sex hormones like estrogen and testosterone, which regulate development and reproduction. Additionally, steroids are converted in the liver into bile acids. These acids are secreted into the intestine to help emulsify and digest the dietary fats we eat. In modern medicine, we have also learned to synthesize steroids. These synthetic versions are widely used as powerful anti-inflammatory drugs to treat severe conditions, ranging from asthma to life-threatening allergic reactions. Having understood their biological roles, let us look at how we represent these complex organic molecules mathematically to understand their mass. The formula shown here allows us to calculate the exact molecular mass of a steroid, like cholesterol. Conceptually, this formula simply states that the total mass is the sum of the masses of all its individual atoms. The variable m represents the total molecular weight. The variables n sub c, n sub h, and n sub o represent the exact count of carbon, hydrogen, and oxygen atoms in the molecule. We multiply each count by their respective standard atomic weights, represented by m sub c, m sub h, and m sub o. Physically, if the number of carbon atoms increases, for example by attaching a longer side chain, the total molecular weight m will increase proportionally, making the molecule even more bulky and hydrophobic. Let us apply this mass formula with a concrete example. Imagine a biochemistry student needs to prepare a cholesterol solution and must know its exact molecular weight. We are given the chemical formula for cholesterol, which has 27 carbons, 46 hydrogens, and 1 oxygen. We chose our summation formula because it perfectly accounts for each element's contribution. In the first step, we substitute the atom counts and the standard atomic weights for carbon, hydrogen, and oxygen. Next, we calculate the intermediate mass contributed by each specific element. Finally, adding these together gives us our final answer of 386.73 grams per mole. This relatively high molecular weight physically confirms why cholesterol is such a large, bulky, and highly hydrophobic molecule. Now that we have calculated the total mass, let us look at how those carbon atoms are distributed across the molecule structure. Because all steroids share the exact same foundational 4-ring structure, we can set up a simple algebraic equation to account for all the carbon atoms. The formula is C total equals C nucleus plus C methyl plus C tail. Conceptually, this breaks the molecule into its structural zones. C total is the total carbon count. C nucleus represents the carbons locked inside the rigid styrene core, which is universally 17. C methyl accounts for any small methyl groups attached to the rings. Finally, C tail represents the carbons in the flexible alkyl side chain. If a steroid has a longer tail, C tail increases, which directly increases C total. Let us use this distribution equation to solve a structural problem. We want to calculate the exact number of carbon atoms present specifically in the flexible alkyl side chain of cholesterol. We are given that the total carbon count is 27, and we know there are 2 methyl groups attached to the core. We use our distribution formula to isolate the unknown tail segment. We substitute 27 for the total, 17 for the universal core, and 2 for the methyl groups. We simplify the right side by adding the core and methyl carbons together, giving us 19. By subtracting these 19 carbons from the total 27, we find our final answer. The alkyl side chain contains exactly 8 carbon atoms. This flexible 8-carbon tail is biologically crucial for anchoring the molecules securely within the cell membrane. We have covered a lot of ground today, so let us summarize the key takeaways. First, remember that steroids are a unique and vital class of lipids, defined not by long, floppy chains, but by their rigid, 4-ring carbon skeleton, known as the styrene nucleus. Second, this compact structure allows them to perform highly specialized functions, most notably stabilizing our cell membranes. Finally, by simply modifying the functional groups and side chains attached to this universal core, nature creates a vast array of molecules, from cholesterol to powerful signaling hormones. Understanding this structure is the absolute key to understanding how they interact so perfectly with the biological systems in our bodies. Great job following along.